Every autumn, the forest floor becomes a graveyard. Leaves fall by the billions, branches crack and tumble, trees that stood for centuries collapse into shadow. Tons of matter, carbon, nitrogen, cellulose, lignin, piling up year after year, century after century. If nothing intervened, the forest would drown in its own debris. The soil would vanish under mountains of the dead. But that doesn't happen. The forest breathes. The soil stays rich. The dead vanish, not into nothing, but into everything else. Something is eating the graveyard. Something is reading every fallen thing, molecule by molecule, and translating death back into life. It's been doing this for 400 million years, long before animals learned to decay, long before bacteria could break down wood. We call them mushrooms, but that's like calling an iceberg ice. What you see fruiting above ground is just the message. The real work, the alchemy, happens in the dark, because mushrooms don't just consume death, they remember it, they store it, they transform it into the code that builds tomorrow's forest. What we're about to uncover isn't just biology, it's the reason nothing on Earth ever truly ends. It's why matter has memory. It's how the planet learned to forget death entirely. This is Omni, stories about life, matter, and the mystery in between. Like the video, drop your thoughts in the comments, and subscribe if you believe curiosity should never end. Picture a single oak leaf, turned bronze by October cold. It detaches, falls, lands on soil darkened by 1,000 autumns before it. Within hours, it's not alone. The leaf has been colonized, not by visible creatures, but by something already waiting in the earth. Fungal threads, thinner than spider silk, have detected it. They've sensed the chemical signature of fresh cellulose, the molecular announcement that food has arrived. These threads, hyphae they're called, begin to grow, not randomly, deliberately. They extend toward the leaf with purpose, following concentration gradients of the compounds leaking from its edges. They're not just growing, they're hunting. Within days, the hyphae penetrate the leaf surface. They slip between cell walls, secreting enzymes that break molecular bonds that have held for months. The leaf's structure, its veins, its photosynthetic machinery, its waxy cuticle, begins to dissolve. But this isn't destruction, it's translation. The fungi are reading. Every carbohydrate, every protein, every trace mineral in that leaf contains information. Where the tree grew, what season it lived through, what nutrients it absorbed from soil and sun. The mycelium doesn't just consume this, it processes it, sorts it, redistributes it through an underground network that connects not just one tree, but dozens, sometimes hundreds. That single oak leaf, which seems to vanish into the forest floor, doesn't end. It becomes glucose flowing through fungal highways. It becomes nutrients traded to tree roots in exchange for sugars. It becomes the building blocks of next spring's growth. The leaf dies, yes, but its matter never does. The mushroom won't let it. This is what mushrooms do. This is what they've always done. We've known about decomposition for as long as we've studied biology. But what we're discovering now goes deeper than rot, deeper than recycling. These aren't just organisms breaking down waste. They're information processors, molecular archivists, memory keepers of everything that's ever lived and fallen. According to research published in Nature in 2023 by mycologist Merlin Sheldrake and his team at Cambridge, fungal networks don't just move nutrients, they store chemical signatures of past decomposition events. When mycelium encounters a familiar plant compound it's broken down before, it responds faster, more efficiently. The network remembers, it learns. Think about what that means. Every autumn for 400 million years, mushrooms have been taking notes, cataloguing what falls, what dies, what can be remade. They've built a library of death and a manual for resurrection. But here's what most people miss. Mushrooms aren't breaking things down. They're translating them, from solid to liquid, from structure to flow, from individual to collective. Death, in the language of fungi, is just matter-changing dialects. And they don't do this alone. The mycelium, the vast underground network that produces mushrooms like a Tree produces fruit isn't just fungal tissue, it's a collaboration. Bacteria live along its surface, processing compounds the fungi can't touch. Microscopic animals graze on both, breaking matter down further. Roots from dozens of plant species plug into the network, trading carbon for nutrients, sending chemical signals through fungal intermediaries. The forest floor isn't a graveyard. It's a computational substrate, a living internet made of decay, where information flows through chemistry instead of electricity, where memory is stored in molecular patterns, where every death becomes data that builds the living world. Scientists call this the mycorrhizal network. Some call it the Wood Wide Web, and the name is more accurate than it sounds. Because just like the internet revolutionized how humans share information, the fungal network revolutionized how forests share resources. Trees that are dying send carbon to younger trees through the mycelium. Parent trees recognize their offspring through chemical signatures and feed them preferentially. Entire groves coordinate their growth, their flowering, their defense against disease, all through messages carried by fungal threads they'll never see. But the network does something else, something we're only beginning to understand. It makes death impossible, not metaphorically, literally. Because in a fungal network, matter never leaves the system. Carbon trapped in a falling leaf returns to a growing root. Nitrogen locked in dead wood flows to sprouting seedlings. The atoms that built one generation become the atoms that build the next. Nothing exits, nothing ends. The forest, connected by mycelium, is a closed loop of perpetual transformation. And it's not just forests. Mushrooms decompose everything. Every grassland, every wetland, every desert ecosystem runs on fungi breaking down the dead and redistributing their components. Without them, life would have choked on its own corpses within a few thousand years. The planet would be sterile rock piled with the mummified remains of everything that ever grew. Instead, Earth is alive, constantly renewed, constantly remembering what it was and rebuilding what it will be. Tonight, we're going to follow matter through death and back out the other side, from the moment a leaf hits soil to the moment its atoms rebuild a flower, from the chemistry of decomposition to the philosophy of what it means that nothing can ever truly end. We'll trace the mycelial network beneath your feet, explore the molecular machinery that reads death like a language, and discover what happens when an entire planet learns to forget endings entirely. Because understanding mushrooms changes everything. It reveals that death isn't an exit, it's a translation, that matter has memory, that the boundary between ending and beginning is just a question of perspective, of time scale, of whether you're watching from above ground or below. Let's begin where all cycles begin, with something falling into darkness. Maybe the forest has always been thinking. We just weren't listening. Let's start with something you can see on any walk through the woods. A fallen log, grey and soft, covered in shelf fungi like stacked dinner plates. You've probably stepped over hundreds of them, barely noticing. But if you stopped, knelt down, pressed your finger into that wood, you'd feel something impossible. The log is solid oak, or it was. Oak is one of the hardest woods on Earth, dense enough to sink in water, tough enough that builders have used it for ships and houses for thousands of years. A living oak's heartwood can resist rot for decades even after the tree dies. And yet here, in the middle of the forest, this massive trunk is soft as cork. You can push your thumb through it, crumble it between your fingers like stale bread. Something turned stone into powder. Something dismantled one of nature's most durable structures, molecule by molecule, and left almost nothing behind. That something is growing on the surface, those shelf fungi, Ganoderma or foams, or any of a dozen species that specialize in wood. But what you see, those rigid fruiting bodies, they're not doing the work, they're just the announcement. The real demolition is happening inside the log, in the dark, performed by threads so thin that thousands could fit through the eye of a needle. Mycelium, fungal hyphae, the invisible machinery of decomposition, and what they're doing isn't simple. It's not like fire-consuming paper, it's precise, surgical, targeted. Because wood isn't just one thing, it's a composite material, engineered by evolution to be nearly indestructible. The bulk of any tree trunk is cellulose, long chains of glucose molecules linked together in crystalline sheets, stacked and cross-braced for strength. Wrapped around that cellulose is hemicellulose, another carbohydrate polymer that acts like mortar between bricks, and surrounding everything, impregnating the entire structure like concrete reinforcement, is lignin. Lignin is the problem. It's the second most abundant organic polymer on Earth after cellulose, and it's a monster. Chemically, it's a three-dimensional aromatic network, a tangle of benzene rings and carbon chains bonded in irregular randomized patterns. There's no repeating structure, no predictable weak points. It's not a molecule, it's a molecular maze, and it's almost impossible to break down. Almost. According to research published in Science in 2024 by biochemist Phil Kirsten at the University of Wisconsin, only a handful of organisms on Earth can fully degrade lignin, and nearly all of them are fungi. Specifically, white rot fungi like Phanerochyta chrysosporium and Tramptes versicolor, species that have evolved enzyme systems so specialized, so chemically aggressive, that they can do what almost nothing else can: crack open aromatic rings and pull apart the linen maze. They do this with peroxidases, enzymes that generate free radicals, highly reactive, molecules that rip electrons away from lignin's bonds. It's molecular violence, controlled chaos deployed with precision. The fungi secrete these enzymes into the wood, flooding the lignin structure with oxidative stress until the bonds shatter and the polymers collapse into fragments small enough to absorb. But here's what makes this extraordinary. Peroxidases are expensive. They require energy, cofactors, metal ions. The fungus has to invest resources to produce them, and lignin itself, it's not nutritious. Breaking it down doesn't feed the fungus directly. Lignin is just the wrapper. The prize is underneath, the cellulose and hemicellulose locked inside. So why bother? Why evolve this incredibly expensive, chemically extreme system just to get at sugars? Because for about 60 million years after plants colonized land, nothing could break down lignin. Trees died and fell, but they didn't decompose. They piled up. Forests became graveyards of standing and fallen wood, layer upon layer, compressing under their own weight. This is where coal comes from. Vast deposits of unrotted plant matter from the Carboniferous period, before fungi learned to eat lignin. The entire planet was choking on its own growth. Then, around 300 million years ago, something changed. White rot fungi appeared. They cracked the code. And suddenly, the graveyard started disappearing. Carbon locked in dead trees began flowing back into the ecosystem. The carbon cycle closed. Life could breathe again. The fungi didn't just adapt to decompose wood. They saved the planet from suffocating under its own corpses. And they're still doing it. Right now, in every forest on every continent, fungi are secreting peroxidases and lacases and manganese peroxidases, generating reactive oxygen species, dismantling lignin molecule by molecule. They're performing chemistry that human industrial processes still can't replicate efficiently. We've tried. Delinification, removing lignin from wood pulp to make paper, requires boiling wood chips in caustic chemicals at high pressure. It's energy-intensive, toxic, expensive. Fungi do it at room temperature in the dark using nothing but enzymes and patience. Once the lignin barrier falls, the rest is easier. Cellulose is just glucose chains, and fungi have cellulases, enzymes that snip those chains at specific points, breaking them into shorter fragments, then into individual glucose molecules. Hemicellulose falls to hemicellulases. Proteins in the wood get cleaved by proteases, lipids, waxes, resins. All of it gets dismantled by a pharmaceutical cabinet's worth of specialized enzymes, each one secreted at the right time, in the right amount, targeting the right substrate. The mycelium isn't just growing through the wood, it's reading it, sensing what compounds are present in what concentrations and deploying the appropriate molecular tools. If the wood is rich in nitrogen, the fungus produces fewer nitrogen-scavenging enzymes. If it's dry, the fungus secretes compounds that retain moisture. If it encounters a patch of wood already colonized by bacteria, it changes tactics, sometimes cooperating, sometimes producing antibiotics to clear the competition. This is active, responsive chemistry. The log isn't passively rotting, it's being systematically disassembled by an organism making decisions in real time about how to allocate its molecular resources. And every molecule that gets broken down doesn't just vanish, it gets absorbed into the hyphal network, Glucose flows through the mycelium as energy. Nitrogen gets incorporated into amino acids and proteins. Phosphorus, potassium, calcium, every nutrient locked in that wood gets liberated and pulled into the fungal body, then redistributed through the network to wherever it's needed most. Some of it stays in the fungus, some of it gets traded to plant roots in exchange for sugars those plants made through photosynthesis. Some of it gets stored in the soil, bound into stable organic compounds that can persist for years, decades, even centuries. The mycelium is not just consuming the log, it's curating it, sorting its components, deciding where each piece goes next. Think about what that means on a forest scale. Every autumn, billions of leaves fall. Every decade, thousands of trees die. Every century, entire groves collapse in storms or disease. That's millions of tons of organic matter, carbon, nitrogen, phosphorus, trace metals, water, everything that took decades to accumulate, all hitting the ground at once. Without fungi, that matter would just sit there, pile up, lock away nutrients. Leaves would stack meters deep. Logs would last for centuries, monuments to the dead that nothing could reclaim. Instead, within months, the leaves are gone. Within a few years, a fallen tree is soft enough to crumble. Within a decade, barely a trace remains. The forest floor stays thin, rich, alive. Because fungi are eating everything, processing everything, translating solid structure back into liquid flow, into gases, into nutrients that can move again. Decomposition isn't decay, it's translation. From one form of matter to another, from tree to mycelium to soil to tree again. The molecules never leave, they just change hands. But breaking down is only half the story. What happens to all that matter once it's dissolved? Where does it go? And who decides? Where does it go? And who decides? If you could shrink down and follow a nitrogen atom from that decomposing oak log, you'd see something remarkable. Something that looks less like decay and more like logistics. The nitrogen, once locked in a protein deep inside the wood, gets cleaved by a protease enzyme. The amino acid floats free, absorbed through the cell wall of a fungal hypha, and then it moves, not randomly, directionally. Through the hyphal network, driven by pressure gradients and active transport, flowing like water through pipes, except these pipes are alive, responsive, constantly rerouting based on demand. That nitrogen might travel meters, tens of meters, sometimes hundreds of meters, passed from hypha to hypha, moving through a network that connects not just the log it came from, but dozens of other logs, hundreds of plant roots, patches of soil scattered across the forest floor. The mycelial network isn't just a fungus, it's infrastructure, a living transportation system that moves nutrients across distances no single organism could bridge. This is the Wood Wide Web, and the name undersells it. Because the internet moves information, the mycelial network moves matter itself. Carbon, nitrogen, phosphorus, water, redistributing resources across an entire ecosystem in real time based on need. Let's follow that nitrogen atom a little further. The hypha it's travelling through is part of a massive network, possibly the largest organism on Earth. In Oregon's Malheur National Forest, researchers discovered a single Armillaria solidips fungus that spans 2,385 acres, nearly 4 square miles. It's at least 2,400 years old, possibly much older. The entire thing is one individual, one genetic entity, one continuous network of branching hyphae that has been growing, exploring, connecting for millennia. And it's not unique. Mycorrhizal networks, partnerships between fungi and plant roots, can span even larger areas. According to research published in Nature Communications in 2023 by ecologist Suzanne Simard at the University of British Columbia, a single forest's fungal network can connect hundreds of trees across dozens of species, creating what she calls a socialist economy of resource sharing. Here's how it works. The nitrogen atom moving through the mycelium reaches a junction, a place where the fungal hypha has wrapped around the tip of a tree root, forming what's called a mycorrhiza, literally fungus root. The structure is intimate, microscopic. The fungal cells don't penetrate the root cells, but they press so close that only a thin membrane separates them. Across this membrane, a trade happens. The fungus offers nitrogen, the tree offers carbon, glucose, sucrose, simple sugars it made through photosynthesis, capturing sunlight and turning it into chemical energy. Both organisms need what the other has. The fungus can't photosynthesize. It lives in darkness, underground, and needs carbon from somewhere. The tree can't efficiently scavenge nitrogen from soil. Its roots are thick, relatively few, limited in reach. The fungal network is everywhere, superfine, with thousands of times more surface area to absorb nutrients. So they trade. Carbon for nitrogen, sugars for minerals, energy for structure. But it's not a simple barter, it's negotiated, dynamic. The tree can adjust how much carbon it sends to the fungus based on how much nitrogen it's receiving. The fungus can preferentially supply nutrients to trees that pay better. If a tree gets stingy, cutting carbon supply, the fungus can throttle nitrogen, delivery, or even redirect resources to neighbouring trees that are more generous. This is an economy with prices, supply chains and market forces, and the fungus is the broker. That nitrogen atom, originally from the decomposing oak log, gets transferred across the membrane into the tree root. From there, it moves up through the xylem, incorporated into amino acids, proteins, chlorophyll. It might end up in a leaf. It might help build new growth. It might get stored for winter. The tree doesn't know and doesn't need to. What matters is that nitrogen from a dead tree 20 meters away is now feeding a living one, delivered through a network the tree itself didn't build and can't see. And that tree isn't alone. The same fungal network is connected to dozens of other trees, different species, different ages, different health statuses, a massive Douglas fir, 500 years old, shading the forest floor, a young hemlock seedling struggling in low light, a birch on the forest edge getting full sun, all of them plugged into the same mycelial grid, all of them trading with the same fungal partners. Simard's research revealed something stunning. The large old trees, she calls them mother trees, send carbon to younger trees through the fungal network, even when those young trees are different species. Not just to their own offspring, though, they do recognise and preferentially feed their genetic relatives through chemical signatures in their roots. They also support the entire neighbourhood, subsidising seedlings that would otherwise die in deep shade. When researchers experimentally shaded young trees, cutting their photosynthesis and carbon production, the trees should have starved. But they didn't. Carbon flowed to them from older trees through the mycelial network, enough to keep them alive until conditions improved. The forest, connected by fungi, was acting like a superorganism, redistributing resources from where they're abundant to where they're needed. But the network does more than just move nutrients, it moves information. When a tree gets attacked by insects, it produces defensive chemicals, tannins, resins, compounds that make leaves less palatable or toxic. That response takes time, energy, biochemical machinery. But here's the strange part. Neighbouring trees, even if they haven't been attacked yet, start producing the same defensive chemicals. They're preparing, preemptively defending against a threat they haven't encountered. How do they know? Through the mycelial network. According to research published in Ecology Letters in 2024 by forest ecologist Tamir Klein at the Weizmann Institute, trees under insect attack send chemical signals through the fungal network, specific volatile organic compounds that trigger defense responses in connected trees. The mycelium isn't just a passive conduit, it's a communication infrastructure, a biological telegraph carrying warning signals across the forest floor at speeds far faster than insects can travel. This happens with drought stress too. Trees experiencing water shortage close their stomata, reduce transpiration, shift metabolism to conserve resources, and trees connected to them through the fungal network start doing the same thing. Even if their local soil moisture is fine, they're responding to information about conditions elsewhere in the network, preparing for stress that might be coming. The forest, through fungi, has become predictive, responsive, coordinated in ways that individual trees, rooted and isolated, could never achieve alone. But if mushrooms are moving all this matter around, coordinating all these exchanges, responding to signals and redirecting resources, are they just passive pipes, biological infrastructure following simple rules, or are they making decisions? That question leads somewhere unexpected, somewhere that challenges what we think we know about minds, about intelligence, about what it means to think at all. Are they just passive pipes, biological infrastructure following simple rules, Or are they making decisions? Let's run an experiment. Take a petri dish, place a block of agar, basically fungal food, at one end, place a mycelial network at the other. Between them create a maze, a series of channels with multiple paths, dead ends, correct routes. Then wait. The mycelium begins exploring. Hyphae extend in all directions, feeling their way through the channels, branching at every junction, testing every path. Some reach dead ends and stop growing. Others keep extending, wandering, searching. The network is spreading like water, finding its level, filling every available space. Then one hyphal tip reaches the food. Contact. Chemical signal. Information flows back through the network. Food here, this direction, this path works. And here's where it gets strange. Over the next few hours, the mycelium reorganizes. Hyphaean dead-end channels begin to retract, pulling resources back. Hyphae along the shortest path to the food thicken, reinforcing the successful route. Within a day, the chaotic exploratory network has simplified into a single, optimized connection. The most direct path from starting point to food source. The fungus solved the maze, without a brain, without neurons, without any central processing unit coordinating the search. This isn't hypothetical. Researchers at Hokkaido University in Japan, led by mycologist Toshiyuki Nakagaki, ran exactly this experiment in 2000, using Physarum polycephalum, a slime mold that behaves remarkably like fungal mycelium. The results were published in Nature, and they were undeniable. The organism not only solved mazes, but did so efficiently, finding near-optimal paths through complex networks. They took it further. They recreated a map of Tokyo, placing food sources at locations corresponding to major cities around the capital. They placed the slime mold at Tokyo's position. The organism grew, exploring, connecting the food sources. Within 26 hours, it had built a network structure that closely resembled the actual Tokyo rail system, one of the most efficient transportation networks ever designed by human engineers. The fungus, with no knowledge of civil engineering, no understanding of topology or graph theory, had independently arrived at the same solution humans spent decades and billions of yen developing. How? By following simple local rules. Each hyphal tip responds to chemical gradients, grows toward food, branches at decision points, and maintains connections that prove useful while pruning ones that don't. No single hypha knows the whole maze. No central controller is optimizing the path. But the collective behavior, the network as a system, solves problems that would require intelligence if a human were doing it. This is called swarm intelligence, or distributed cognition. Intelligence without a brain. Computation through growth. Problem solving as an emergent property of many simple units following local rules and communicating through chemical signals. But here's what makes fungal networks even more remarkable. They don't just solve problems, they remember solutions. According to research published in PNAS in 2023 by mycologist Lynn Boddy at Cardiff University, when mycelial networks encounter a nutrient source they've exploited before, they respond faster the second time. The network remembers where food was found and adjusts its growth pattern accordingly. The memory isn't stored in any particular location. It's distributed, encoded in the architecture of the network itself, in the thickness of certain hyphal connections, in the chemical gradients that persist even after growth moves on. They tested this by presenting fungi with food sources in specific patterns, say, food every 4 centimeters in a straight line. After several repetitions, they removed the food but kept the spacing. The mycelium continued growing as if the food was still there, extending hyphae at 4 centimeter intervals, anticipating resources that had been present before but weren't anymore. The fungus had learned a pattern. It was predicting, expecting, behaving as if it had formed an internal model of its environment and was acting on that model even when reality had changed. That's not stimulus response. That's memory informing behavior. That's learning, and it gets stranger. When researchers stressed mycelial networks, exposing them to salt, heavy metals, or toxins, the networks learned to avoid those stresses in future encounters, even if the chemical signature was slightly different. They generalized from experience. If a fungus encountered copper toxicity in one area, it would avoid not just copper, but other heavy metals with similar chemical properties, treating them as potential threats based on prior learning. This is categorization, abstraction, the ability to take specific experiences and extract general principles that apply to new situations. We usually associate these capabilities with nervous systems, with neurons forming synapses, strengthening connections through repeated activation, storing information in the patterns of neural networks. But fungal mycelium does something analogous with entirely different hardware. Instead of electrical signals crossing synaptic gaps, chemical signals diffuse through cell walls. Instead of neurons, there are hyphal tips communicating through molecular gradients. But the functional result, learning, memory, adaptive behavior, is the same. Philosopher Andy Clark would call this morphological computation. The structure of the network is the computation. Growth patterns encode information. The shape of the mycelium reflects its history of interactions with the environment. You can read a fungal network like a map of everything it's encountered, every nutrient source it's found, every threat it's avoided, written in the branching architecture of its body. But if fungi can learn and remember, what exactly are they remembering? And for how long? Consider what a mycelial network experiences over its lifetime. Every fallen leaf it decomposes, every root it connects with, every drought, every flood, every seasonal cycle. That's decades of information for a typical forest fungus, centuries for old-growth networks, millennia for the largest individuals. And unlike animals, which replace neurons gradually, maintaining continuity of self through constant cellular turnover, fungi grow indefinitely. The hyphae that connected trees 100 years ago might still be functioning, still carrying nutrients, still part of the living network. There's no clear boundary between past and present structure. The fungus is its history. Its body is a physical record of every place it's been, every resource it's found, every successful strategy it's employed. In a very real sense, the mycelial network is a memory bank, not metaphorically, literally. It stores information about past decomposition events, past nutrient locations, past environmental conditions in the persistence of its structure and the chemical gradients it maintains. When that network encounters a familiar leaf type, say, oak, it responds differently than when encountering an unfamiliar species. The enzyme profile shifts. Certain celluloses get upregulated. Lignin-degrading peroxidases deploy faster. The fungus isn't starting from scratch. It's accessing something like procedural memory, a knowing how embedded in its biochemical response patterns. Researchers at the University of Exeter demonstrated this in 2024 by tracking gene expression in Pleurotus austriatus, oyster mushrooms, as they decompose different substrates. When presented with beechwood, which the species commonly encounters, the fungus activated specific enzyme cascades within hours. When given tropical hardwood it had never encountered, the response was slower, more exploratory, less efficient. But the second time it encountered that tropical wood, even weeks later after growing on other substrates, the response was faster. The network had updated its library, stored the solution, remembered. This raises a question we don't usually ask about organisms without brains. What is the subjective experience, if any, of a fungal network? When mycelium solves a maze, what is it like to be the mycelium? Is there anything it's like at all? We can't know. Consciousness remains one of science's deepest mysteries, and attributing it to fungi based on problem-solving behavior would be premature. But what we can say is this: whatever intelligence is, whatever computation is, it doesn't require the biological hardware we thought it did. It doesn't require neurons or brains or centralized processing. It can emerge from decentralized networks of simple units, exchanging chemical information and adjusting their structure based on feedback. The forest floor is thinking, not with words or images, not with consciousness as we experience it, but with chemistry and growth and memory encoded in form. The mycelium is solving optimization problems, learning from experience, coordinating behavior across vast distances, making decisions, resource allocation, growth direction, which trees to feed, which compounds to produce, based on information accumulated over timescales that dwarf human lifetimes. If that's not intelligence, it's something functionally indistinguishable from it. But this ancient partnership does more than just move nutrients and solve mazes. it creates something impossible, biological immortality. And to understand that, we need to go back, way back, to a time before forests, before trees, before anything green had ever grown on land, way back. To a time before forests, before trees, before anything green had ever grown on land. 450 million years ago, Earth's continents were barren. Rock, sand, mineral dust sculpted by wind and water, but no soil, no grass, no flowers, no trees. Life existed, thrived even, but it was all underwater. The oceans teemed with bizarre invertebrates, early fish, and vast forests of algae. But the land was dead, a sterile planetary surface baking under unfiltered sunlight, because there was no ozone layer yet, nothing to shield the surface from UV radiation intense enough to shred DNA. And then something happened, something that would change the planet forever, that would transform those barren continents into the green world we know. But it wasn't plants that did it. Not at first. It was fungi. According to fossil evidence analysed by paleobotanist Linda Graham and published in Science in 2010, the oldest undisputed land fossils aren't plants. They're fungal spores and hyphal structures preserved in rocks from the Ordovician period, roughly 460 million years ago. Fungi colonised land before plants did. They were the pioneers, the first complex organisms to figure out how to survive in an environment that had never supported life before. But they didn't stay alone for long, because shortly after fungi appeared on land, plants followed, not coincidentally. The two arrived together, or more accurately, plants arrived because of fungi. The oldest plant fossils, simple bryophytes resembling modern liverworts, show something remarkable: fungal hyphae inside their tissues, not attacking them, supporting them. The very first land plants were already mycorrhizal, already partnered with fungi. This makes sense when you understand what plants were attempting. Moving from water to land isn't a simple transition. Water provides constant hydration, dissolved nutrients, structural support through buoyancy. On land, there's none of that. A plant has to solve dehydration, find nutrients in mineral substrate that barely qualifies as soil, support its own weight against gravity, and do all this while exposed to intense solar radiation. Early plants had no roots. They had rhizoids, simple hair-like structures that could anchor them but couldn't efficiently absorb water or nutrients. They couldn't grow tall because they lacked vascular tissue. They couldn't spread far because spores were their only reproductive strategy, and land environments were harsh, unpredictable, frequently lethal to unprotected cells. Fungi solved most of these problems. The hyphal networks acted as surrogate root systems, extending far beyond what the plant could reach, absorbing water and nutrients from a huge area and delivering them to the plant in exchange for sugars the plant made through photosynthesis. The fungal partner provided drought resistance, access to minerals, even some protection from UV radiation through pigments in the hyphal walls. Without fungi, plants wouldn't have colonized land. Life would have stayed in the oceans. The continents would still be barren rock. There would be no forests, no grasslands, no terrestrial ecosystems at all. No insects, no reptiles, no mammals, no birds, certainly no humans. Fungi didn't just enable the greening of Earth, they caused it. They are the reason land became habitable, and the partnership has never ended. Today, over 90% of land plant species form mycorrhizal associations. From the smallest moss to the tallest redwood, from tropical rainforest to Arctic tundra, plants and fungi are locked in a mutualism so ancient, so fundamental, that neither can exist in most environments without the other. But fungi did more than just help plants colonize land. They invented decomposition. And that invention reshaped the carbon cycle, the atmosphere, the climate, everything. For about 60 million years during the Carboniferous period, roughly 360 to 300 million years ago, forests spread across the continents. Massive lycophyte trees, some over 30 meters tall, ferns the size of modern oaks, early conifers, all growing, thriving, dying, and falling, and not rotting. Because lignin had evolved, plants had figured out how to build woody tissue, structural material strong enough to grow tall to compete for sunlight to dominate ecosystems. But nothing had yet figured out how to break it down. When those trees died, they just accumulated. Fell on top of each other, layer after layer, forming organic deposits meters thick. The coal we burn today? That's unrotted Carboniferous forest. Hundreds of millions of years of plant growth that never decomposed because fungi hadn't yet evolved the enzymes to dismantle lignin. The planet was locking away carbon at a catastrophic rate. Atmospheric CO2 levels plummeted. Oxygen spiked to 35%, the highest in Earth's history, enabling giant insects, dragonflies with 70-centimetre wingspans, millipedes 2 meters long, because oxygen diffusion could support such large exoskeletons. The climate destabilized. Ice ages began. The whole system was out of balance because the carbon cycle was broken. Carbon kept exiting the atmosphere into dead plant matter, but nothing was returning it. Life was choking on its own success. Then, around 290 million years ago, white rot fungi appeared. The fossil record shows the sudden presence of lignin-degrading fungi in rock layers from that period, and shortly afterward, coal deposition slowed dramatically, then nearly stopped. Trees still died and fell, but now they decomposed. Carbon locked in wood began returning to the atmosphere. The cycle closed. According to research by geobiologist Colin Ward published in Geobiology in 2023, the evolution of fungal lignin degradation stabilised Earth's climate, prevented runaway glaciation, and allowed forests to persist without burying the planet under their own debris. Fungi once again saved the terrestrial biosphere, this time by inventing the recycling system that keeps it running, and they've been refining that system ever since. Over 300 million years, fungal decomposition has become astonishingly efficient, specialised, diverse. Different fungal species target different substrates. Some specialise in fresh leaf litter, attacking soft tissue and simple carbohydrates. Others focus on woody debris, deploying the heavy enzymatic machinery needed for lignin. Some prefer animal remains, breaking down keratin, chitin, bone. Some even specialise in specific tree species, co-evolving with their hosts so intimately that certain fungi only decompose certain trees. The result is a global decomposition network operating continuously in every terrestrial ecosystem, processing every bit of organic matter that dies. Leaves fall in autumn and vanish by spring. Dead trees collapse and soften within years. Animal carcasses, insect bodies, shed skins, discarded exoskeletons, all consumed, dismantled, returned to circulation. Without fungi performing this service, the planet would be unrecognisable. Imagine the forest floor buried under centuries of unrotted leaves, meters thick, impenetrable. Imagine fallen trees lasting for millennia, creating impassable tangles of dead wood. Imagine nutrients locked away in corpses, unavailable to the living. Plant growth would slow, then stop, as nitrogen and phosphorus became sequestered in the dead. New growth could only happen where catastrophic events, fire, flood, landslides, physically broke down organic matter through brute force. Life as we know it, the verdant, thriving, constantly renewing biosphere, exists because fungi figured out how to undo death faster than death could accumulate. But calling it undoing death isn't quite right, because nothing is being undone. Matter isn't being destroyed or erased, it's being transformed, translated, moved from one state to another. The atoms that built a tree don't disappear when the tree dies. They don't even leave the forest. They just change form, from solid to dissolved, from structure to flow, from individual to collective. And here's the truly strange part. Because matter never leaves, because fungi recycle everything, the forest becomes functionally immortal. Trees die, but their atoms live on, incorporated into new growth, new trees, new generations. The carbon in a young sapling might have been in a tree that fell a century ago. The nitrogen in its leaves might have cycled through a dozen organisms since then, fungus to bacteria to plant to insect to fungus again, round and round, never exiting the system. If death is just transformation, and matter remembers, then what does that mean for endings, for permanence, for the idea that anything can truly be lost? This raises a question we don't usually ask. If matter never dies, does it remember what it used to be? If matter never dies, does it remember what it used to be? Let's track a single carbon atom. Call it C12, the most common stable isotope. Right now, it's in the trunk of an old Douglas fir, locked inside a cellulose molecule in the heartwood, 30 metres above the forest floor. It's been there for 70 years since the tree was young, since that section of trunk was growing sapwood conducting water and nutrients upward. The tree absorbed that carbon atom as CO2 from the air. A leaf, somewhere in the canopy, opened its stomata one morning in spring, let air diffuse in, and a molecule of carbon dioxide drifted into the mesophyll tissue. There, inside a chloroplast, light energy split water molecules and drove electrons through photosystem II and I, generating ATP and NADPH. Those molecules powered the Calvin cycle, a series of enzyme-catalyzed reactions that stitched CO2 into a 3-carbon sugar. That sugar moved through the phloem, was converted to cellulose by enzymes in the cambium, and got incorporated into the growing cell wall of a xylem cell that would eventually become heartwood. For 70 years, that carbon atom stayed put, locked in structure, immobile, part of the tree's permanent scaffolding. Then the tree dies. Lightning strike, bark beetle infestation, old age. Doesn't matter. The tree falls. The trunk hits the forest floor with an impact that shakes the ground, crushing ferns, breaking smaller trees, creating a sudden gap in the canopy where light floods down, and the carbon atom waits, still locked in cellulose, still part of a massive molecular structure that won't break down on its own, not for decades, possibly centuries if conditions are dry enough. But conditions aren't dry. This is a Pacific Northwest forest. Rain falls constantly, the log wicks moisture into its tissues, and within weeks, fungal spores that have been dormant in the soil detect the chemical signature of fresh cellulose. They germinate. Hyphae extend, following concentration gradients of lignin breakdown products, volatile organic compounds that signal food. Within months, dozens of fungal species have colonized the log. White rot fungi attack the lignin barrier. Brown rot fungi follow, targeting cellulose once the lignin scaffolding weakens. Soft rot fungi work the surface, exploiting the moist outer layers. Each species deploys specific enzymes, occupying specific ecological niches within the decomposing wood. A white rot fungus reaches the cellulose molecule containing our carbon atom. It secretes cellulose, an enzyme that binds to the cellulose chain and cleaves the glycosidic bond linking glucose units together. The long polymer fragments into shorter chains. Other cellulases attack those fragments, breaking them down further. Eventually, individual glucose molecules float free, released from the matrix that held them for 70 years. The carbon atom, now part of a free glucose molecule, gets absorbed through the cell wall of a fungal hypha. It crosses into the cytoplasm, and then it moves. Through the hyphal network driven by cytoplasmic streaming, the active flow of cellular contents, the glucose molecule travels, not far at first, maybe a few centimetres, to a growing hyphal tip that needs energy for elongation. There, enzymes break the glucose apart through glycolysis and the citric acid cycle, extracting energy stored in its chemical bonds. Our carbon atom ends up in a CO2 molecule. The fungus respires it, expelling it through the hyphal wall back into the soil air. 70 years in wood, a few weeks in fungus, and now it's free again, gas diffusing through soil pores. But it doesn't escape. A root, a few millimetres away, is respiring too, pulling in oxygen, expelling CO2. The local concentration of carbon dioxide in the soil is higher than in the atmosphere above. Diffusion would normally carry the CO2 upward, but before it can rise far, another root tip, this one from a young hemlock seedling struggling in the shade of the fallen Douglas fir, absorbs it. The carbon atom, now CO2 again, enters a stomata on a needle, gets fixed by rubisco in a chloroplast, enters the Calvin cycle, becomes part of a glucose molecule again. That glucose flows through the phloem to the root tip, where it's traded to a mycorrhizal fungus in exchange for nitrogen. Nitrogen that same fungus scavenged from decomposing leaf litter 2 meters away. The carbon atom is now in the fungal network again. Different species this time, a mycorrhizal partner rather than a decomposer, but the same basic structure, hyphae, cytoplasmic streaming, cellular respiration. The fungus uses the glucose for growth, extends its network, encounters another root, trades more nitrogen for more carbon, and our carbon atom, it gets incorporated into the cell wall of a hyphal tip. Becomes structural again, locked into chitin, the polysaccharide that fungi use instead of cellulose. It stays there for a few years as the hyphal network persists. connecting dozens of trees, moving nutrients, growing, retracting, responding to seasonal changes. Then the hemlock that fed this particular fungus dies. Too much shade, too much competition, not enough resources to sustain it. The mycorrhizal association breaks down. The fungal hyphae in that area stop getting carbon from the tree and begin to die off. Bacteria move in, decomposing the dead hyphae, breaking down the chitin, liberating the carbon atom once again. It gets incorporated into bacterial biomass, becomes part of a bacterial cell wall. That bacterium gets eaten by a soil nematode. The nematode digests it, uses some of the carbon for its own metabolism, excretes the rest. The carbon atom ends up in nematode feces, a tiny pellet of organic matter in the soil. That pellet gets colonized by more fungi. The cycle repeats. Carbon back into mycelium, back into CO2, back into a plant, back into the soil, round and round. This is what ecologists mean when they talk about nutrient cycling. But cycling sounds clean, mechanical, like a factory conveyor belt moving resources in predictable paths. The reality is messier, more chaotic. That carbon atom doesn't follow a simple circle. It spirals, ricochets, bounces between organisms and pools of dead organic matter, sometimes moving quickly, sometimes getting locked away for years in stable compounds before something liberates it again. But here's the key insight. It never leaves. Through all these transformations, tree to fungus to air to plant to fungus to bacteria to animal to fungus again, the carbon atom stays in the forest. It cycles between living biomass and dead organic matter and soil and air, but the system is closed. Functionally, if not perfectly, the forest recycles itself. According to research published in Global Change Biology in 2024 by ecosystem ecologist Cara Allen at the University of British Columbia, forests in equilibrium, mature forests where growth equals mortality, have residence times for carbon averaging 50 to 200 years. That means a carbon atom entering the system stays, on average, for centuries before escaping to the atmosphere or being carried away in stream water. During that time, it might cycle through dozens of organisms, be incorporated into structure, and respired back out, locked away and liberated over and over. The forest is a slow-motion whirlpool, pulling matter in and holding it, circulating it, internally, releasing it only gradually. And fungi are the mechanism that makes this work. They're the recyclers, the loop closers, the reason matter doesn't just accumulate as debris but flows back into the living system. Now scale this up. Not one carbon atom, but all of them. All the carbon in all the trees, all the nitrogen in all the proteins, all the phosphorus in all the DNA, all the minerals in all the tissues. Billions of tons of elements moving through the forest ecosystem, cycling between organisms, between living and dead, between structure and flow, and it's been happening for millions of years. The atoms in your body right now, carbon, nitrogen, oxygen, phosphorus, have almost certainly been through this cycle before, multiple times. That carbon in your hemoglobin might have been in a dinosaur once, in a fern before that, in a fungus before that, in decaying wood before that. Traced back far enough, every atom in every organism has passed through countless other organisms, been incorporated and released and incorporated again in an unbroken chain stretching back to the origin of life. You're made of recycled matter. Everyone is. Everything is. There are no new atoms, no virgin materials, only ancient ones, older than multicellular life, older than Earth itself, formed in the nuclear furnaces of stars that died before our solar system existed, now circulating through ecosystems, through bodies, through the same loops that have been running for billions of years. This is biological immortality, but not the way we usually think about it. Not individuals living forever. Individuals die. Always. Cells wear out. DNA accumulates damage. Entropy wins. But matter doesn't die. It can't. Conservation of mass forbids it. Atoms persist, get rearranged, incorporated into new structures but never destroyed. And fungi are the custodians of this process. The translators, the agents that take matter locked in death and make it available to life again. Without them, the cycle breaks. Matter gets sequestered, nutrients become unavailable, the system grinds to a halt under the weight of its own corpses. With them, death becomes impossible, not as an event, organisms still stop functioning, still cease to be what they were, but as a finality. Because the matter that made up that organism continues, flows onward, becomes something else, gets remembered in a sense by being incorporated into the next generation. But if death is just transformation, and matter remembers, then what does that mean for us? For the way we think about endings, about loss, about what it means to be temporary beings made of permanent atoms. But if death is just transformation, and matter remembers, then what does that mean for us? Here's a strange fact. Scientists can tell what you ate for breakfast by analysing your hair, not from residue or particles stuck to the strands, but from the isotopic signature of the atoms themselves. Carbon-13 and nitrogen-15 ratios vary. Depending on diet, more meat means higher nitrogen. 15, more corn-based products mean higher carbon-13 because corn photosynthesizes differently than other plants. These ratios get incorporated into the proteins your body builds, including keratin in hair. Months later, even after you've forgotten that meal, your hair remembers. Not metaphorically, chemically. The atoms carry information about their history embedded in their isotopic composition. This works for anything. Archaeologists use it to reconstruct ancient diets, tracking what Neolithic humans ate by analysing bone collagen from skeletons thousands of years old. Ecologists use it to trace nutrient flow through food webs, following nitrogen isotopes from soil bacteria to fungi to plant roots to herbivores to predators. Forensic scientists use it to identify human remains, matching isotopic signatures to geographic regions where people lived based on the water they drank, the food they ate. Matter remembers where it's been, what it's done, what processes it's passed through. According to research published in Nature Geoscience in 2023 by geochemist Naomi Levin at the University of Michigan, you can reconstruct entire paleo environments, ancient climates, vegetation types, rainfall patterns, from isotopic signatures preserved in soil carbonates and fossil teeth. The ratios of oxygen-18 to oxygen-16, carbon-13 to carbon-12 tell stories about temperature, aridity, the types of plants that dominated ecosystems millions of years ago. The atoms themselves are archives. Every isotopic ratio is a data point encoding environmental conditions at the moment that atom got incorporated into biological tissue or mineral structure. And those ratios persist, sometimes for millions of years, carrying information forward through time. This isn't memory the way we think about it. Neurons storing patterns of electrical activity, brains reconstructing experiences from synaptic weights. This is chemical memory, structural memory, information encoded in the physical properties of matter itself, and fungi interact with this constantly. When mycelium decomposes a leaf, it's not just breaking down carbohydrates and proteins, it's reading chemical signatures, isotopic ratios, trace element compositions, secondary metabolites that vary by species and environmental conditions. That oak leaf has a different chemical fingerprint than a maple leaf, different from a birch leaf, different from last year's oak leaf because rainfall was lower and nutrient availability changed. The fungus detects these differences, responds to them, adjusts its enzyme production, its growth rate, its resource allocation based on what it's consuming. In a sense, it's reading the leaf's history, where it grew, what stresses it experienced, what nutrients it contained, written in molecular language. Research by biochemist Katharina Pawlowski at Stockholm University, published in Molecular Ecology in 2024, demonstrated that mycorrhizal fungi can distinguish between root exudates from different plant species, different individuals of the same species, even different genetic lineages within populations. The fungi are detecting chemical variations subtle enough to encode identity, who this plant is, what family it belongs to, whether it's kin or stranger, and they act on that information. Mycorrhizal fungi preferentially allocate nutrients to plants that are better partners, those providing more carbon, those with compatible chemical signalling. They recognize kin through root exudate chemistry and support relatives over non-relatives. This is nepotism, favoritism, discrimination based on chemically encoded identity. The fungus is reading molecular memories, genetic signatures expressed as metabolites, and making social decisions based on that information. But here's where it gets philosophically strange. If atoms carry information about their past, if matter remembers what it's been through, then every organism is partly an archive. Your body contains carbon that was once in other organisms, nitrogen that cycled through soil bacteria, oxygen that came from photosynthesizing algae. Those atoms carry traces, faint isotopic signatures, elemental associations of where they've been. You're made of memories that aren't yours. Molecular memories, chemical histories written in the very substance of your cells. And when you die, those memories don't disappear, they get read. Fungi and bacteria decomposing your body will process every molecule, sort every atom, redistribute everything you were made of back into the ecosystem. Some of your carbon will be respired to the atmosphere, some will be incorporated into fungal biomass, some will be traded to plant roots, some will be locked into soil organic matter for decades or centuries. But the isotopic signatures will persist. The ratios of carbon-13 to carbon-12, nitrogen-15 to nitrogen-14, oxygen-18 to oxygen-16, those won't change through decomposition. They'll get diluted, mixed with atoms from other sources, but they'll remain. Scattered through the ecosystem, your atomic identity will persist, a chemical ghost readable by any instrument sensitive enough to detect it. This is material continuity, the opposite of oblivion, not consciousness surviving death. Consciousness almost certainly doesn't, requiring functional neural networks that decomposition destroys, but matter surviving. And matter that carries information, that remembers, that can be traced back through cycles and transformations. Ecologist Robin Wall-Kimmerer, in her work on indigenous perspectives in ecology, describes this as a gift economy. When you die, you give your atoms back. They become gifts to the soil, the fungi, the plants, the next generation. Nothing is lost because loss implies something leaving the system. Everything stays. Everything continues. But Western culture largely rejects this continuity. We build monuments trying to preserve individual identity against decay. Embalming, metal caskets, concrete burial vaults, all designed to slow decomposition, to keep the body intact, to resist the transformation that fungi offer. We treat decomposition as destruction, as the ultimate loss, the final indignity. What if we've had it backward? What if decomposition is the opposite of loss? It's reintegration. The body returning to the ecological community it was always part of, matter rejoining the cycles that built it in the first place. Mycologist Merlin Sheldrake conducted an experiment documented in his book Entangled Life and subsequent research published in 2024. He created a living coffin, a burial pod seeded with fungal mycelium designed to accelerate decomposition and integrate human remains directly into forest ecosystems. The mycelium breaks down the body rapidly, thoroughly, turning tissue into fungal biomass and soil nutrients within months instead of years. The fungi then fruit, producing mushrooms that release spores, spreading the network further. Early results show dramatically faster nutrient integration compared to conventional burial. Carbon and nitrogen from the body enter the soil system within a season, become available to plants within a year. From a chemical perspective, the person becomes the forest. Their atoms flow into trees, flowers, insects, birds, everything connected to the mycelial network. This isn't metaphor, it's measurable atomic redistribution. You can track it with isotopic tracers. The person as matter persists, not as consciousness, not as memory or personality or anything we'd recognize as self, but as substance, as elements, as atoms that once assembled into thoughts and are now assembling into leaves, roots, wings, soil. Is this immortality? Not individually. You, the pattern of information encoded in your neural networks, the subjective experience of being you, that ends. But you as matter, that continues indefinitely. The atoms reading this sentence right now will exist long after you stop being able to read. They'll become something else, many things actually, scattered through the ecosystem, incorporated into countless organisms over centuries. And here's the deepest strangeness. This has already happened to you multiple times. The atoms you're made of have been in other organisms before. Thousands, probably millions of organisms over billions of years. Every carbon atom in your body was once in something else. Ancient bacteria, Cambrian trilobites, Jurassic ferns, Pleistocene megafauna. Your matter has been alive countless times before, in countless forms, none of which you remember. You're not your atoms. You're a pattern, a specific arrangement of matter, a configuration that maintains itself for a few decades through constant molecular turnover, then dissolves. But the matter continues, gets rearranged into new patterns, new organisms, new configurations of increasing complexity or degrading simplicity depending on entropy and energy flow. Fungi are the mediators of these transformations. They're not creating or destroying, they're translating, taking patterns written in dead tissue and rewriting them into living networks, into soil structure, into nutrients that build new organisms. They're editors, revisers, translators working in the language of chemistry, turning endings into beginnings by refusing to recognize the distinction. From a fungal perspective, if we can imagine such a thing, there are no individuals, only flows. Carbon flowing from air to plant to soil to fungus to air again. Nitrogen flowing from atmosphere to bacteria to plant to animal to fungus to plant again. The boundaries we draw between self and other, between living and dead, between individual and environment, are arbitrary designations we impose on continuous processes. The mycelial network doesn't respect those boundaries. It connects everything, moves matter across what we think of as divisions. A tree dies, but its carbon feeds a dozen living trees through fungal bridges. An animal dies, but its nitrogen builds proteins in plants meters away. Nothing ends, everything transforms. If death is just transformation, and matter remembers, then what does that mean for endings? For permanence? For the idea that anything can truly be lost? Maybe loss is the wrong word. Maybe what we call death is just matter changing address. Atoms relocating from one structure to another, information being translated from one substrate to the next. The fungi know this, have known it for 400 million years. They've been reading molecular memories, translating chemical histories, facilitating transformations the entire time life has existed on land, and they're still doing it. Right now, beneath your feet as you read this, consuming, processing, redistributing, making death impossible by making it indistinguishable from continuation. If death is just transformation, and matter remembers, then what does that mean for us? For the way we think about loss, about endings, about what happens when we vanish? Maybe it means we've been asking the wrong question, not what happens after death, but what doesn't. Maybe it means we've been asking the wrong question, not what happens after death, but what doesn't. We began by watching the forest floor become a graveyard every autumn. Leaves falling by the billions, branches cracking, trees collapsing, matter piling up year after year enough to bury the world. But it never does. The graveyard empties itself, not through disappearance, but through transformation. And we know now what's doing the work. Fungal networks stretching through soil, reading every fallen thing, translating death back into the vocabulary of life. This isn't cleanup. It's not waste management or decay. It's computation, memory, redistribution. It's matter flowing through cycles so old they predate forests themselves, so fundamental that life on land exists only because these cycles do. The mushroom fruiting on a fallen log isn't consuming the tree, it's continuing it, taking structure and turning it into flow, taking stasis and returning it to circulation. The tree's atoms don't end when the tree dies. They just become available again for roots, for leaves, for insects, for bacteria, for soil, for everything connected to the network. And the network connects everything. Through fungal threads thinner than thought, the entire forest communicates, trades, remembers. Trees feed their offspring through mycelial highways. Plants recognize kin through chemical signatures. Organisms separated by meters or decades exchange resources through fungal intermediaries that forget nothing, that carry molecular memories forward through time. We've spent centuries drawing boundaries between species, between individuals, between life and death. Clean categories that make the world comprehensible, that let us say, this is me, that is other, this is alive, that is dead. But fungi dissolve those boundaries. show them to be arbitrary lines drawn across continuous processes. Where does the tree end and the fungus begin when their cells are pressed so close together that molecules flow between them without clear transfer points? Where does death occur when the matter that built an organism continues functioning, flowing, participating in life processes after the organism stops? Where is the individual when your survival depends on networks you didn't build, partnerships you can't see, organisms you'll never know? These aren't rhetorical questions. They're challenges to our basic ontology, our categories of existence. And fungi have been posing them for 400 million years, since they first formed partnerships with primitive plants on ancient shorelines, enabling the invasion of land. Everything green you see, every forest, every grassland, every flowering plant, exists because fungi invented mutualism on a planetary scale. Everything decomposable returns to circulation because fungi invented the enzymatic machinery to dismantle lignin, cellulose, chitin, everything life builds. Everything connected, trees to trees, plants to plants, species to species, connects through networks fungi engineered long before neurons existed. Life on land is fundamentally mycological. Not plant-based, not animal-based, but fungal-based. The visible organisms, trees, flowers, us, are expressions of underground architecture we mostly ignore. Networks doing the real work of moving nutrients, storing information, coordinating ecosystems. We live on the fruit of fungal labour, walk on soil they built, breathe air balanced by carbon cycles they regulate, eat food grown in fertility they maintain, and when we die, we return to them. Not as loss, not as ending, but as contribution. Matter rejoining the pools it came from, atoms continuing their journeys through forms we can't predict. This should change how we think about death. Not eliminate the grief, loss of specific patterns, specific relationships, specific configurations of matter we call people still hurts, still matters. But maybe it contextualizes that loss differently. You end, but your matter doesn't. The atoms that assembled into your thoughts will assemble into something else. Many things, actually. Trees and fungi and bacteria and soil and eventually, inevitably, other people. The carbon in your body has been alive before, was in ancient organisms you'll never know about, participated in life processes billions of times over billions of years. It's alive now, as you, and it will be alive again, in forms neither of us can imagine, after you stop being you. That's not consolation exactly. Consciousness probably doesn't survive. Pattern dissolves when substrate breaks down. But it is continuity. Material continuity, atomic persistence, the impossibility of true endings in a universe where matter is conserved. And fungi are the mechanism ensuring that continuity. The translators, the recyclers, the network engineers, making sure nothing gets lost, nothing exits the system, nothing fails to return. They've been doing this since before trees existed, before animals walked on land, before flowers bloomed or grass spread or any of the familiar forms of terrestrial life appeared. They were here first, partnered with the earliest land plants, enabled everything that came after, and they'll be here after us, still processing, still translating, still making death impossible by making it indistinguishable from transformation. There's humility in this. We're not separate from these cycles, not observers above natural processes. We're in them, made by them. Our intelligence, our consciousness, our entire existence depends on planetary networks we didn't build and can't control, maintained by organisms most people never notice. But there's also beauty. The forest doesn't mourn its dead because death doesn't exist in the way we think it does. Trees fall, but their atoms rise again in seedlings. Leaves decay, but their nutrients feed next year's canopy. Nothing is lost because loss requires things to leave. And here, in the fungal economy, nothing leaves. Everything stays, circulates, returns. Maybe that's what the mushrooms have been teaching us all along, that endings are perspective dependent, that death is taxonomy, not ontology, a category we invented, not a natural boundary. That matter remembers what it's been and carries that memory forward into what it will become. We started by asking why nothing really dies. Now maybe we see, it never needed to. The question was wrong from the beginning. Death isn't something that needs to be prevented or overcome or explained away. It's transformation. One state becoming another. pattern dissolving so matter can be repurposed, recycled, remembered. The mushroom growing on that fallen oak isn't killing it. The tree was already dead. The mushroom is reading it, translating it, making it available, turning ending into, beginning by refusing to see them as different processes. And if you walk through a forest in autumn, when leaves are falling and mushrooms are fruiting and the whole system is preparing for winter, you're not walking through death. You're walking through transformation, through matter in motion, through cycles so ancient, so fundamental, that they predate thought itself. The forest floor isn't a graveyard. It never was. It's a library, an archive, a memory bank where every fallen thing gets read and remembered and rewritten into what comes next. And the mushrooms are the librarians cataloguing everything, forgetting nothing, ensuring that every atom that ever lived keeps living, in form after form after form forever. We began by watching things fall into darkness. Now we understand. They weren't falling. They were returning, coming home to networks that never forgot them, that held space for their atoms, that would translate them back into life the moment they arrived. Nothing ends. Nothing can. The mushrooms have known all along. They've been teaching the same lesson for 400 million years, in every forest, on every continent, beneath every footstep. Death is transformation. Matter is memory. And the network, patient and invisible, waits to receive everything that falls, not as loss, but as gift, not as ending, but as beginning again.